Hi there, welcome to the last lecture in chapter 9. So I'm going to try and finish this up. We only have a few slides left. So we're still talking about how to count cells. And so the last method is an indirect method where we're not actually counting cells, but we're counting the amount of material produced by cells. And this is known as turbidity. So turbidity is the amount of materials within a solution, and if it's a very turbid solution, light can't penetrate. If it's a less turbid solution, light will penetrate. And so we can use a spectrophotometer, and this sends a ray of light. That ray of light will then hit the solution, and if it can go through the solution is not considered very turbid. If it's reflected slightly, that would mean very little bit of turbidity. But in this case, we have all this light coming in and it gets reflected right away. Nothing makes it through to the detector, or very minimal makes it through to the detector. And what that tells you is that this is a very turbid solution. The more turbid, the more estimate of cells that we have, a higher estimate of cells, okay? All right, so now let's look at some requirements that bacteria need. So when we're looking at prokaryotic organisms, bacteria as well as archaea, they can inhabit almost every environment on Earth. I would say that they inhabit all environments. But most that we are aware of inhabit areas that humans also can inhabit. But there are some that are considered extremophiles that inhabit areas where humans can't really inhabit. We know of some of them because we have had researchers go out and test these areas. And I'm sure that there are some areas where we have never tested because we just can't get there. And so there's some bacteria that probably live in areas where we've never been. So we don't know every organism on Earth. And so those organisms would be called extremophiles. So the things that we're going to look at is the atmosphere. So what is the amount of oxygen that organisms can utilize? pH, do they need a more acidic or an alkaline environment? Temperature, what temperature can they grow really effectively at? And then how much water do they actually need? Or can they survive in? All right, so the first thing is oxygen. We have 5 different groups of organisms based on oxygen requirements. The first is called the obligate aerobe. And so this is a really nice picture that shows each group of organisms. This would be that fluid thioglycolate solution that you guys tested bacteria in. And if you remember, we had obligate aerobes and we had facultative anaerobes. But this is all 5 groups. So obligate aerobes are organisms that require oxygen. They cannot survive without oxygen. And though oxygen is a very toxic agent, they have special enzymes that protect them from the toxicity of oxygen. We then have obligate anaerobes, so organisms that cannot grow in the presence of oxygen. Like I just said, oxygen is a very toxic molecule. And so there are organisms, in fact, the first organisms that lived on Earth were anaerobic or anoxygenic. They could not survive in the presence of oxygen. And so they grow at the bottom of the tube because they can't grow in oxygen. And then we have those facultative anaerobes. Facultative anaerobes can grow with or without oxygen, but they prefer oxygen. They grow better in oxygen. We also have aerotolerant anaerobes. These are organisms that do not use oxygen, but they can survive in the presence of oxygen because they have those enzymes that protect them from the reactive oxygen species, so those free radicals that oxygen can form. And last but not least, we have microaerophiles. These are also known as capnophiles. So they grow at a low level of oxygen. And because of that, they have to have oxygen, but a lower level. they grow down below. So there's the most oxygen is going to be at the very top where oxygen from the air can diffuse in. They're going to grow down below about 15% lower than our obligate aerobes will grow. They're called capnophiles because they often grow in the presence of more carbon dioxide than oxygen. So I talked about oxygen being a dangerous molecule, and it is dangerous, but even though it's dangerous, it's necessary for the growth of certain organisms, including humans. And so there are 3 enzymes that are produced by organisms that are oxygen users: peroxidase, superoxide dismutase, and catalase. And what they do is they break down reactive oxygen species, or they bind to, so keep reactive oxygen species, free radicals, from damaging our cells. And so we can detect different ones using different tests. And the test that we use to detect for if you had the enzyme, or if you have catalase present, we used a catalase test, and we took hydrogen peroxide and put it onto our bacteria. If there was bubbling, that meant that catalase was breaking down the hydrogen peroxide, which is toxic to our cells, and producing water and oxygen as a byproduct. There's also a pH factor that microbes grow best in. So again, most bacteria that we know about are going to be neutrophiles. They grow best at a more neutral pH, whereas acidophiles grow at a lower pH than neutral. Acidophiles are going to grow optimally at a pH that's below or near 3.0. We have examples of acidophiles, your lactobacillus, which is part of your normal flora. We also have Helicobacter pylori, which grows in your stomach. It's not part of what we would call our normal flora, but it's found in a lot of people's stomachs, and it grows at a very low pH, closer to 1, between 1 and 2, depending on how much hydrochloric acid is in your stomach at any given time. And so this is going to, or these organisms aren't going to grow as effectively if they're in other pHs. And then we have alkylophiles, which are organisms that grow at a higher pH. So they're going to grow above pH of 8. An example would be Vibrio cholera. This species causes the disease cholera, and which causes severe diarrhea and dehydration. So we have some extremophiles, both extreme acidophiles and alkylophiles, that are going to grow effectively at their own extreme pHs, but they will not grow really at all at a normal neutral pH. So here we have acidophiles. If you grow over here at the 1, you're probably not going to grow very effectively at a 7. But there are some that can grow at a 3, and they might be able to grow even, they might grow slightly around at the 6. Neutrophiles are going to go anywhere from 5.5 to about 8.5, but they grow best at this pH of 7. And then we have alkylophiles that have a range as well that overlaps the neutrophiles. So there's no big overlapping here, but that doesn't mean that an acidophile couldn't grow at a pH of 6. It's not going to grow as effectively at a pH of 6, okay? There's always exceptions to every rule. So optimum pH is just where they grow the best at. So I want to show that again. Organisms also grow and thrive at different temperatures. So there are some organisms that are called psychrophiles. Psychrophiles grow at very cold temperatures, but they can also grow at close to room temperature, depending. Then we have organisms that are mesophiles. Mesophiles grow best at body temperature, but they can also grow at a lower temperature. We have thermophiles that can grow at a high temperature that's slightly higher than body temperature, and they can grow up to about 80 degrees Celsius. And then we have the hyperthermophiles, which are those organisms that grow at the most extreme temperatures. So again, psychrophiles grow best at low temperatures. So 0 to 15 degrees Celsius. 20 degrees Celsius is about room temperature. So this would be a cold environment, but something that you could still survive in. Whereas psychrotrophs thrive between 4 degrees and 25 degrees Celsius. So slightly warmer in both cases. These are going to be organisms that are going to be found in your refrigerator, that are going to be spoilage microorganisms. They're going to, your fungi that grow on your food and cause all kinds of problems with your food. That's what you're going to see here. Then we have the mesophiles that grow anywhere between, again, around room temperature. I think room temperature is actually 21 degrees Celsius, but I'm not 100%. It really depends on how cool or warm you like your rooms. To about 40 or 45 degrees Celsius, but the optimum is about 20 or 37 degrees Celsius, which is body temperature. Most of our human pathogens are mesophiles. Thermophiles and hyperthermophiles grow at very warm environments. And so they don't tend to cause damage to humans, but they're still very important to humans. We use thermophiles and hyperthermophiles in different research that we do. Last but not least, we have organisms that are called halophiles. So halophiles are going to be organisms that can withstand different osmotic environments. So if you're a halophile, a halophilic organism, then you're able to grow in the presence of a lot of salt and very little water. So normally in the presence of high salt, you would dehydrate, but halophilic organisms can sometimes grow, and even sometimes they thrive in that environment. So they have mechanisms to kick out the salt and keep water inside of them. If you're a halotolerant organism, these organisms can grow and multiply in high salt, but they don't require it. Whereas extreme halophiles require the salt to grow. And an example of a halophilic organism, again, Vibrio cholera, which causes cholera, is an organism that is considered a halophile. So the species that we typically look at, I should have said 4 because we do have a Vibrio species. But this semester, our Vibrio has not been growing as effectively. So hopefully in the future it will grow better. But we have Staph aureus, Staph epidermidis, and Micrococcus luteus that grow really well in the salty environment. Last thing I want to talk about, and we've already covered this, so I'm going to go through this relatively quickly, is different types of media. So in a lab, we grow our microbes on different media. And so the media that we use is going to be dependent on what we're trying to do. So complex media is a media that can grow lots of different organisms, and we can use broth or agar. So a broth is a liquid, agar is that solid media. If you are trying to grow organisms for a research project and you need to know exactly what type of organisms you're growing, you would use a chemically defined medium so that you know exactly what the organisms are getting, and you can then take things away if you want to see which organisms can still grow. An enriched medium is one that has all the basic essential nutrients, but also has added in things that will allow specific types of organisms to grow. Selective media are media that favor the growth of one type of organism and inhibiting another. And then we have differential media, which helps us to separate organisms based on color changes of either the colony or the media itself. And so an example of a selective media, which we used in our lab, is phenyl ethyl alcohol agar. So this is a selective media. It is selective for the growth of gram-positive organisms, specifically our staphylococcal and streptococcal groups. And then it inhibits gram-negative organisms. The phenyl ethyl alcohol inhibits gram-negative bacteria from growing. Then we have selective and differential media that we use. If it's both selective and differential, we always look at the selective first and then the differentiation. So MSA, mannitol salt agar, this is a selective media that allows for the growth of halotolerant organisms. So first it selects for halotolerance. And then if you grow, it separates you based on your ability to grow or your ability to utilize mannitol. So it selects for halotolerance and then separates based on mannitol usage. Here's a mannitol user, here's a non-user. We also have eosin methylene blue agar. Eosin methylene blue agar is selective and differential. This time it selects for gram-negative bacteria. And methylene blue and eosin are both inhibitory of gram-positive. And then those that do grow are all gram-negative, and then it will differentiate them based on their ability to utilize lactose. And so here we have a lactose-negative gram-positive, or we have a lactose-negative gram-negative bacteria. Here we have a lactose-positive gram-negative bacteria, turned purple. And then if you get this bright green metallic color, that is indicative of an extreme lactose fermenter. All right, that is all I have for you. So I'm going to stop here and I will upload this video in just a little bit. Bye.